Welcome everybody to my presentation, the gut-brain link with autism spectrum disorders, which I prepared especially for the conference focused on autism in inclusive education. And firstly, I want to thank to Professor Dr. Joanna Kosewska for inviting me to participate on this online conference. And also, I would like to send warm greetings to the leader of this project, Professor Dr. David Priest from University of Northampton. We in MSSA, Macedonian Scientific Society for Autism, and also we in Macedonia greatly support such a kind of conferences. At the very beginning, I want to introduce you in my lecture. Autism spectrum disorder is a range of neurodevelopmental disorders that affect communication and social behavior. Besides social deficits, systemic inflammation, gastrointestinal immune-related problems, and changes in the gut microbiota compositions are characteristic for people with ASD. Majority of children with ASD display a lot of gastrointestinal symptoms, which we will see in the next slides. And also there is a lot of evidence about intestinal permeability. There are several hypotheses point to environmental influences as possibly being causative to autism, including involvement of abnormal gastrointestinal microbiota composition, autoimmunity, early environmental exposures to viruses, and drug compounds. Also, there is growing evidence suggests that the gut microbiota has a role in the pathophysiology of autism spectrum disorders. What is microbiota? Microbiota is a collective term for this microbial community, which includes bacteria, archaea, eukaryotes, and viruses. There is a degree of variation in the adult composition of gut microbiota, and this can be influenced by diet, antibiotic usage, lifestyle, and genetics as well. The microbiota is crucial for health in humans with several important metabolic, protective, and trophic functions. Changes in the gut microbiota seen in ASD may have a causative role and perpetuate gastrointestinal symptoms, or may simply be a confounder driven by dietary restriction. Some of recent studies have suggested that alterations in the gut microbiota composition in children with ASD may contribute to both gastrointestinal and neurological symptoms as well. Probably you will ask me what is the aim of this presentation. The aim is to show possible mechanism of gut-brain axis in the pathophysiology of ASD and to find possible treatments for this condition. Here on this slide you can see the connectivity between gut and brain, and some of the authors says that our gut and our gastrointestinal system is our second brain. There are a lot of influence on neurotransmitters, stress, anxiety, mood, behavior, because the same neurotransmitters interact in our gut and in our brain. We will see on the next slides that connection. Now I will want to stress your attention about gut-brain axis. What is that? There are some studies which confirm a gut-brain nervous system link in autism, which opening a new direction in the search for potential treatments that could ease behavioral issues associated with autism by targeting the gut. The nervus vagus or vagus nerve connects the gut and brain. It is a 10th nerve from our cranial nerves, and it is the longest nerve in our body, and it is the connection between, because some of its branches are in our gut, it is the connection between our gut and the brain. This gut-brain axis provides us a greater understanding of the connection between diet and disease, and it is also relevant in a lot of other psychiatric disorders, not only in the ASD. The human microbiome, which has co-evolved with humans, is a community of different bacteria that inhabit the body and is beneficial to humans. One of the study reveals a gene mutation that affects neuron communication in the brain and was the first identified as a cause of autism, also causes dysfunction in the gut. On this axis, the mechanisms and interactions of sensory cues and biochemical signals that take place between food, the gastrointestinal tract, and the nervous system can best be described. There is a link between autism gene and the gut brain. The study reveals a gene mutation that affects neuron communication in the brain and was the first identified as a cause of autism, also causes dysfunction in the gut. This mutation affects communication by altering the velcro between neurons that keeps them in close contact. Researchers in the gut-brain axis team have built on this clinical work with a series of studies on the function and structure of the gut in mice that have the same velcro gene mutation. It was found that this mutation affects gut contractions, the number of neurons in the small intestine, the speed that food moves through the small intestine, and responses to a critical neurotransmitter important in autism, well known in the brain but not previously identified to play any major role in the gut. Now I want to talk a little bit about gut-brain axis and the microbiome. High rates of comorbidity between gastrointestinal and psychiatric illness exists in the literature. Bidirectional communicative and regulatory system involving but not limited to the brain and central nervous system and the enteric environment of the gut. Messages sent received by gut and brain via the enteric nervous system through neural pathways such as the efferent sympathetic system and the afferent vagal nerve, as well as through the bloodstream. Gut and its microbiota affect immunity, endocrine function, neural development, cognition and behavior, as well as regulation of behavior. All of these systems are affected in the children and persons with ASD. Stress and the accompanying arousal affect the gastrointestinal function in top-down and bottom-up signals via the enteric nervous system. Here on this picture are shown some of this explanation that a very complex link between our gut microbiota and in the gut, and also connection with the blood vessels through the cytokines, tumor necrosis factor, interleukins, and the other, which I will talk lately. What is the human microbiome? The microbiome is defined as the collective genomes of the microbes composed of bacteria, bacteriophage, fungi, protozoa, and viruses that live inside and on the human body. According to Young Joy, the Human Microbiome Project, extending the definition of what constitutes a human according to National Human Genome Research Institute. And on this picture, you can see where is our microbiome located. And it is under our genetics, environment, diet, lifestyle, hormones, and also industry. And we can find in our nose, mouth, lungs, stomach, colon, sexual organs, and our skin as well. We all know, probably also you who will listen and watch this presentation, know about a lot of gastrointestinal abnormalities. According to the Hollingue author et al. In 2017, they published most common gastrointestinal issues are constipation in 22%, diarrhea in 23% of ASD persons, abdominal pain, discomfort, which have 14%, bloating, flatulence 13%, nausea, vomiting around 6%, and any other symptoms 47%. Also, the big findings are in food sensitivities, preferences, mealtime behaviors, and toileting problems are very common in those children with ASD. Here on this picture are shown that sometimes children have esophagitis, gastritis, lactose intolerance, dysmotility of the gut, microbiota dysregulation, minimal colitis of the colon, diarrhea or constipation. It depends. Some children have diarrhea, some have constipation. There is no rule. Dysphagia, nausea, and a lot of heartburn because of esophagitis, and a lot of other symptoms are very, very common in the children with ASD. What is in the gastrointestinal pathology in ASD? There are pathologists who find infiltrated helper and cytotoxic T cells and CD19 B cells in biopsies of the duodenum, terminal ileum, and colon of autistic patients with gastrointestinal disturbances. Enhanced density of dendritic T cells was observed in the colon of ASD children with gastrointestinal disturbances compared to histologically non-inflamed controls, and even compared to controls suffering from lymphoid nodular hyperplasia, Crohn's disease, and ulcerative colitis as well. A factor that might contribute to the gastrointestinal disturbances among autistic individuals is abnormal composition of gut microbiota. The data of gastrointestinal disturbances, such as changes in gut microbiota and T-cell infiltration, strongly indicate an altered immune status in the intestine of autistic individuals. On this picture, we can show that gastrointestinal pathology in ASD is very complex, and that gut-brain link is very complex. There is some alterations in the gut, which we call that leaky gut. Probably you have heard about this symptom. Then through that leaky gut, through this lumen of the gut, a lot of ingested allergens and changed microbiota cross into the bloodstream, and there are before that there are a lot of activated T cell lymphocytes, TNF alpha, tryptase, histamine, and also other cytokines who are activated in our bloodstream, and then they cross the blood-brain barrier and cause a lot of changes in our microglia, astrocytes, and neurons as well. Gut microbiome alterations are implicated in a lot of articles in ASD, children and adults. Species which have been reported to change include Clostridium, Desufubrio, Lactobacillus, and Sacharella. Altered gut microbial composition may cause disruption of the gut barrier, potentially allowing translocation of bacteria and their antigens, toxins, and metabolites. Bacterial fermentation of dietary carbohydrates normally leads to the production of short-chain fatty acids. These are signaling molecules with a variety of functions. The most abundant short-chain fatty acids are acetic acid, butyric acid, and PPA. Specific strains produce harmful toxins such as lipopolysaccharides, which can theoretically lead to the production of inflammatory cytokines and impair neurodevelopment. Vargas and some other authors show that there is intestinal inflammation in the body of children with ASD. Chronic inflammation in the gut can damage the epithelial cell layer. When present, this may explain the increased intestinal permeability, which we call leaky gut syndrome, which is found in ASD patients. There is one study of intestinal biopsies among 52 regressive autistic children, which revealed significantly increased CD3 plus and CD3 plus CD8 plus in the epithelium. These are cells of our immune system, as well as CD3 plus in the lamina propria. This is one of the layers of gut, compared with developmentally normal non-inflamed control groups, reaching levels similar to inflamed controls. There is upregulation of pro-inflammatory cytokines, which I mentioned the slide before, in the intestinal mucosa of autistic children, with GI symptoms has also been reported in the literature. Increasing cytokine levels were associated with more impaired communication and aberrant behaviors. Now I want to stress your attention about the role of serotonin, which is neurotransmitter and mediator of inflammation. The serotonergic system has been implicated in the pathogenesis of ASD since increased levels of blood serotonin were first described in children with autism. Around 1/3 of the patients with ASD has blood hyperserotoninemia, 30 to 35% of persons. On the other hand, the capacity of PET hydroxytryptamine synthesis, PET hydroxytryptamine is serotonin, in the global brain was decreased in children with autism. The cause of ASD-related hyperserotoninemia is thought to arise from genetic or immune changes in the body. During an inflammatory response in the gut, 5-hydroxytryptamine is produced and released by enterochromaffin cells and intestinal inflammatory cells such as mast cells and platelets, resulting in a faster moving gut and an increase in secretion, vasodilatation, and vascular permeability. On this slide, I will show you evidence about abnormal intestinal permeability. There is some damage to tight junctions of the gut mucosa, and showed that in some patients with infantile autism, damage to these junctions occurs in the absence of established gastrointestinal disorders. De Magistris and his colleagues replicated the finding of abnormal intestinal permeability in around 37% of 90 autistic children, as well as in 21.2% of their 146 siblings. ASD patients on a reported gluten-free, casein-free diet had significantly lower intestinal permeability values compared with those on an unrestricted diet and controls. Intestinal permeability was found to be involved in the etiopathogenesis of several autoimmune diseases, including Crohn's disease, celiac disease, and type 1 diabetes. Significantly lower levels of the health-promoting bifidobacteria species and the mucolytic bacterium Akkermansia muncinifila were found in children with autism. Now I want to show you the connection between food allergy in ASD. One of the first authors which shows that is Sandra Lucarelli from Rome, Italy, in 1995. In food allergy, presentation of food antigen leads to the response by T cells and subsequently to an initiation of a food-mediated immune response. Parental reports have shown a significantly great incidence of food allergy in ASD patients compared with healthy contrasts. What found Sandra Lucarelli in 1995? Oral challenge with cow's milk protein lead to worsening of some of the behavioral symptoms of autistic children, and that these children express significantly higher serum levels of IgA, IgG, and IgM for casein and IgA for lactalbumin and beta-lactoglobulin compared with healthy controls. Sabra et al. Hypothesized the food allergy is the pivotal causative factor that produces lesions in the ileum that consist of enlarged lymphoid nodules containing large collections of lymphocytes in the gastrointestinal lymphoid tissues adjacent to Peyer's patches. These gastrointestinal lesions would allow the entry of food antigens across the inflamed mucosa of the bowel and elicit an inflammatory response in the gastrointestinal tract. Now I want to compare our study with other study we conducted in my PhD from 1999 until 2002. We established the study similar to Sandra Lucarelli and some American authors about plasma concentration of IgG4 subclass immunoglobulins, which we found that is significantly higher in persons with autism compared to their siblings and to their mothers and fathers. The concentrations of IgA, IgM, total IgG, and subclasses IgG1, IgG2, and IgG3 are not significantly different between autistic children and their brothers and sisters. Plasma concentration of immunoglobulins wasn't significantly different between males and females with autism in our study 18 years ago. Statistically significant higher plasma concentrations of IgG antibodies against alpha-lactalbumin, beta-lactoglobulin, and casein was found in autistic persons compared to their parents. And you can see the P is very low, which is a strong statistically significant difference. And the concentrations of IgG gliadin antibodies in autistic persons was found to be higher compared to their mothers and fathers, which is statistically significant as well. With these findings, we confirmed the findings of Sandra Lucarelli conducted in 1995 study. Plasma concentration of specific allergy IgA antibodies against beta-lactoglobulin in persons with autism is significantly higher compared to brothers and sisters in Macedonian study. The level of casein IgA antibodies in autistic groups was significantly higher compared with those of their mothers and fathers, and also brothers and sisters. The levels of casein and beta-lactoglobulin specific allergic IgE antibodies in the serum of autistic patients was significantly higher compared with those of their brothers, sisters, and their parents. And also, finally, on our Macedonian study, the level of total IgE antibodies was statistically significant higher in autistic persons compared to their mothers and their brothers and sisters. With this study, we confirmed that there is connection between our immune system and central nervous system, and that a lot of children with ASD have immunologic disturbances in their body. What is the association between ASD and maternal allergic diseases? There is a lot of evidence about correlating to the importance of allergy in the pathophysiology of ASD, in the finding that mothers diagnosed for asthma or allergies such as atopic eczema and rhinitis during the second trimester of the pregnancy had a greater than twofold elevated risk for ASD in their offspring. There was an enhanced association observed between allergic conditions and autism in families with more than one ASD affected child. This observation suggests that the genes underlying atopy may be related to the etiology of ASD. The author King hypothesized that epigenetic disruption of brain development is caused by gestational exposure to allergy-associated inflammatory mediators, for example, interleukin-6 and histamine. These mediators promote retinoic acid and estradiol gene transcription, resulting in overexposure of the fetus to retinoic acid and estradiol. Other immune processes in ASD. Although many studies support the hypothesis that ASD is associated with Th2 skewed immune response, there are also studies that indicate the involvement of other immune pathways. Plasma levels of interleukin-12 and gamma interferon were shown to be increased in autistic individuals, suggesting rather an enhanced Th1 response instead of Th2. Reduced cytotoxic activity of natural killer cells was also suggested in the literature. Paul Ashwood and his collaborators reported increased plasma levels of heterogeneous group of cytokines, including interleukin-1 beta, interleukin-6, and many others, making it even more difficult to identify a specific type of immune response. Macrophage migration inhibitory factor, MIF, which is also constitutively expressed in brain tissues, was enhanced in peripheral blood of autistic individuals compared to typically developing controls, and the high plasma MIF levels were positively correlated to autistic behavior. Neuroimmune interactions at the site of the blood-brain barrier. I mentioned something before, now I will explain more. Immune cells produce all kinds of substances upon gastrointestinal inflammation, such as cytokines and chemokines. These immune cells and their substances are not restricted to the gut, but enter the circulation and will therefore pass all organs in the body, including the brain. The brain is highly vascularized organ, but brain cells are protected from harmful compounds in the blood by means of the blood-brain barrier. This barrier is a layer of endothelial cells cemented together with tight junctions. The cells lack intracellular fenestrations and have very little ability to undergo pinocytosis process. Although ASDs are considered neurodevelopmental disorders, the neuropathology remains poorly understood. Brain growth abnormalities are the most prominent findings in the neuropathology of ASD. The brain undergoes a period of rapid growth followed by slow growth later in development. Now I want to tell you a little bit about nutritional interventions which can help in some of the persons with ASD. Many parents report that their children, their autistic child, suffers from gastrointestinal symptoms. This had led to research of the prevalence and characteristic of gastrointestinal disturbances, which we show before. on this presentation, and the contradictory results on the prevalence of gastrointestinal disturbances are likely due to different facts: interpretation of gastrointestinal symptoms, social and communicative impairments of patients, and the heterogeneity of ASD. The severity of autistic behavior was shown to correlate with gastrointestinal disturbances, increased intestinal permeability, and enhanced serum LPS, cytokine, and chemokine levels. Immunomodulatory dietary interventions such as polyunsaturated fatty acids, shortly PUFA, and pre or probiotics, allergen-free diets, and pharmaceuticals, mast cell stabilizers, and anti-inflammatory or immunosuppressive drugs for the treatment of gastrointestinal inflammation could be also beneficial for the treatment of autistic behavior. You probably know about gluten-casein protein-free diets. There is a lot of evidence in the literature about good effects of this diet. A regularly proposed treatment to reduce food-related effects in autism is a GFCF diet. Many non-peer-reviewed articles, books, and websites support and encourage the application of this diet. And also, we in Macedonia, we have seen a lot of benefits from this diet. One research among 284 autistic children indicates that approximately 15.5% of autistic children in the USA use this diet as a complementary treatment. This is not the main treatment. I don't to be understand wrong from you, but this is a complementary treatment which can help in the other behavioral treatments in special education and other methods in treatment and rehabilitation of ASD. There have been many reports on the role of GFCF diet on alleviating several symptoms of autistic individuals. Significant improvements have been noted within psychological and behavioral categories in vocal and non-vocal communication, attention and concentration, episodes of aggressiveness, affection, motor skills, sleeping patterns, displaying of routines and rituals, anxiety, empathy, and responses to learning. The introduction of the GFCF diet is derived from the opioid excess theory of autism, which was explained before by Professor Karl Reichelt, by Professor Paul Shattuck and other researchers. And also in my PhD studies, some of this was proved. On this slide, you can see the top 10 anti-inflammatory foods which you can give to the children or persons with ASD who have elevated antibodies on casein, on gluten, on lactalbumin, and you can see the improvements: onion, garlic, pepper, fruits, and other salmon and other foods. Dietary pre- and probiotic supplementation is also very important in treatment, in biomedical treatment of children with ASD. Probiotics are live microorganisms which, when administered in adequate amounts, confer a health benefit of the host. They are supposed to give relief from lactose, maldigestion, reducing the related episodes of diarrhea. Probiotics are suggested to help reduce risk factors associated with inflammatory bowel disease, colorectal cancer, and impaired gut-associated immune responses. Prebiotics are non-digestible food ingredients that have a beneficial effect through their selective metabolism in the intestinal tract. A reason for a potential influence of prebiotics on the treatment of some symptomatology of autism concerns especially the selective stimulation of growth of lactobacilli and bifidobacteria and the production of SCFA that have an influence on gut energy metabolism, gut barrier function, water fluxes, and motility. One of the biomedical treatments is vitamin supplementation, which I use in my everyday medical practice, and a lot of parents showed improvements of their children. A survey among physicians showed that multivitamin supplements are among the most widely recommended medical interventions for autism and are recommended for children with autism by almost half of the intervened physicians. The available studies involving vitamin mineral supplementation of autistic individuals generally lead to slightly promising results. Nutritional high-dosage supplementation with ascorbic acid resulted in a statistically significant reduction in autism severity. There have been many double-blind placebo-controlled studies of very high dosages of vitamin B6 and magnesium, and all but one showed positive behavioral improvements. The autistic subjects improve in levels of vitamins, minerals, and biomarkers of sulfatation, higher levels of free and total sulfate, methylation, higher levels of S-adenosyl methionine, and lower levels of uridine. Glutathione, higher levels of reduced glutathione, and oxidative stress, higher reduced oxidized glutathione ratio, and lower levels of nitrotyrosine compared with the control group. The role of B vitamins. They are essential for methylation, transsulfuration, folate cycle in our cells, required for neurotransmitter processing, depleted by stress, increased sugar intake, OCP, required activation by gastric acid, and B12 deficiency reduces NMDA receptor activity. Optimizing outcomes. Chemical imbalance causes symptoms. What is the root cause of chemical imbalance? Addresses core underlying problems, inflammation, nutrition deficits, gut dysfunction, mitochondria, and genetics as well. And expand the toolkit, drug therapy plus talk therapy or speech therapy, improved outcomes. Drug therapy plus speech therapy plus nutrition therapy plus lifestyle change can further optimize outcomes. And now I'm finishing my presentation. What are the conclusions of my presentation? ASD is increasingly common condition that affects millions of families across the world. Although there is substantial evidence that the gastrointestinal tract and the gut-brain axis have a central role in autism, further studies are required to understand the etiology and the mechanisms by which these factors act, and to understand the possible cause-effect relationships between food choice and autism. Details regarding antibiotic use, diet, and gastrointestinal symptoms are key to unraveling the link between autism and altered gut microbiota. Diet and nutritional support can modulate epigenetic triggers, which are explained in the etiopathophysiciology of autism spectrum disorders. And the most useful medications in the biomedical treatment of ASD are B vitamins, magnesium, glutathione as a very strong antioxidants, omega-3 fatty acids, probiotics, and selective serotonin reuptake inhibitors. I would like to thank you to all of you who will see and listen this presentation. These are my contacts. If you have some questions after seeing this presentation, please don't hesitate to contact me on my telephone, fax, email. This is my blog website, on Facebook, Twitter profile. You can follow me LinkedIn and also on Instagram, where I publish a lot of about autism, biomedical treatment, and thank you all for your attention. And once again, Best regards from Macedonia. Bye bye.